The first session of the summit will have two important scientists from University of California, San Francisco. You will be processed in English. And the second half of the summit will have two important scientists from Zhongshan Hospital, Fudan University, and the Taichung Mandarin Hospital. So it will be processing Mandarin. Now, I would like to invite the chairman of the Infection Control Society of Taiwan, Professor Chen Yijun. She is also the director of Infectious Disease Division in National Taiwan University Hospital. She will give us an opening speech for this summit and introduce our first speaker, Professor Charles Chiu. Please welcome Professor Chen.
Thank you and welcome everyone to join this international summit. It is my honor to give this opening remark from three perspectives regarding the metagenomic NGS. First, as an ID physician, as infection control specialist, we tracing the potential pathogen causing infection in human or causing outbreak. The second, during the past 20 years, antimicrobial resistance is a global crisis. Among several elements to solve this crisis, the first and most important is diagnosis. The third, in the context of COVID-19, we know the rapid and accurate diagnosis facilitate or control a pandemic. So through these three very important reasons, I really appreciate every one of you to join this international summit. In addition, we would appreciate the people from different medical society in Taiwan, as well as a distinguished speaker from international, including Taiwan, give a wonderful talk, and we anticipate a wonderful summit today. Now, it is my honor to introduce our first distinguished speaker from United States, Professor Charles Chiu. Professor Chiu is Professor of Lab Medicine, Division of Infectious Disease at University of California, San Francisco. He also Director of UCLA Arboviral Diagnostic and Discovery Center and Associate Director of UCSF Clinical Microbiology Lab. You may look at his very long list and a very distinguished performance in the past years. He also have many funding support. Therefore, he had more than 1,000 very good academic publication as well as many patents. But we would like to pay attention regarding his background. He graduated from UC Berkeley, electronic and computer science. He also received complete training for medicine, for infectious disease. He also is a PhD of biophysics. You can understand why he has such good performance today. Let's welcome Professor Charles Chiu. Let's welcome.
Thank you, Dr. Chen. And I would like to thank Taichung General Hospital and the organizers and the Medical Societies of Taiwan for inviting me to speak with you today. Give me 1 second. I want to make sure that my screen, fantastic. Great, so the title of my talk today is called Metagenomic Sequencing for Diagnosis of Infections, SARS-CoV-2 as a Priority Case. So we'll be discussing today some of our work involving clinical metagenomic sequencing for diagnosing infectious diseases. And we'll talk about how the SARS-CoV-2, the COVID-19 pandemic, has really impacted and our ability to do metagenomic sequencing. So this is the first slide that I have. And the reason I want to present this slide is I view clinical metagenomic sequencing as a tool that can be used to help diagnose infections in the hospital. And ultimately, the goal of this technique is to be able to implement it into the clinical laboratory. the concept behind metagenomic sequencing of all of the DNA and RNA in a sample, is very attractive because it provides the promise of having a single test that could be used to diagnose a wide variety of different infections. And if you couple that with the bioinformatics, with the analysis, the goal is that this could be developed in a clinical laboratory as a laboratory developed test that could be used to provide critical information that can guide physicians, information involving the diagnosis and early treatment for patients who are critically ill in the hospital. Like any healthcare test, like any microbiological or clinical test, ultimately the goal is to lower healthcare costs and to improve patient outcomes. And many of you may be familiar with the use of sequencing. The one point with sequencing of infectious diseases is that the turnaround time is absolutely critical. We hope to have to be able to run this test in a matter of hours to days versus days to weeks. So the general concept of metagenomics is that because all of these microbes that you see here are inherently consist of DNA or RNA, in principle, there's no reason why we can implement a single shotgun sequencing method to be able to diagnose all of these infections. And indeed, nearly all microbes, nearly all microorganisms can be uniquely identified by metagenomic next-generation sequencing. This includes bacteria, viruses, fungi, and parasites. We're only missing one group of infectious disease agents, prions, which are protein-based, but the vast majority of organisms can be detectable using a single test. And what we've done at University of California, San Francisco is that we have developed and validated in a licensed clinical laboratory under CLIA regulation an assay or a test called the mNGS, the Metagenomic NGS test. This has been validated for spinal fluid, and my colleague, Dr. Michael Wilson, will be discussing that in much more detail. But we've also developed it for plasma as well and for other body fluids. And this particular test is coupled with an automated bioinformatics pipeline, and we call this pipeline SERPI. It stands for Sequence-Based Ultra-Rapid Pathogen Identification. The general idea is what we want to do is we want to take the raw sequence reads, which can be per run can be up to 400 million reads, roughly 10 to 20 million reads per sample, process them in an automated fashion by removing human reads and then aligning these reads to microbial reference databases. We use GenBank. And then we can identify the presence of viruses, bacteria, fungi, or parasites. This software is also accompanied with a web-based interface. This is a graphical visualization interface that provides an automated results report, a heat map, as well as a coverage map. And this can be used by laboratory physicians who don't need to have bioinformatics expertise. Laboratory physicians and pathologists are able to read out the result and report the result in the electronic medical record. So the clinical utility of this approach was demonstrated in a study that we published in the New England Journal of Medicine a few years ago. This is called the Precision Diagnosis of Acute Infectious Diseases Study. The idea behind this study is this is a multi-institutional study involving 8 hospitals in California nationwide. What we did is that we enrolled in consented patients prospectively. These are all patients who have a neurological illness, which was undiagnosed at the time of enrollment. And what we wanted to do is we wanted to compare head-to-head the diagnostic yield of clinical metagenomic next-generation sequencing testing from spinal fluid against all microbiological testing. The conventional testing that was done on these patients was extensive, including antigen testing, PCR, culture, and serology. And in addition to this, what we implemented was the concept of a clinical microbial sequencing board, very similar to kind of the virtual teleconferencing boards that we have nowadays because of the COVID pandemic. The idea behind the board is that the information is provided in real time, the results, and they're then discussed directly with the treating physician. You can read about the study if you're not already familiar with it, and I give you the reference in the lower left. But one of the highlights of this particular study was that we found that as a single test, metagenomic next-generation sequencing, or mNGS, was as sensitive or more sensitive than conventional testing, direct detection spinal fluid testing combined. And looking into this in a little more in-depth, you can see that roughly among this very difficult to diagnose patient population, 50% of the patients ultimately had a diagnosis. If you look at the part of the pie, the 28% that were infectious diagnoses, there were actually three types of infectious diagnoses. One was these were diseases, infectious diseases that were only identified using the metagenomic mNGS test. There were 19 cases that were diagnosed by both conventional testing of mNGS, and then you had 26 cases that were clinical false negatives by metagenomic sequencing. If we look at the cases that were missed by metagenomic sequencing, they fell into three categories, as you can see in the upper right. The first were cases of diseases which are primarily diagnosed by serology. So for instance, an example of that would be neurosyphilis, which is typically diagnosed by positive antibody testing and not by direct detection testing. There were also some cases where the diagnosis was not made from spinal fluid. It was made from a different tissue type, typically a brain biopsy. Notably, in the majority of those cases, when we subsequently did metagenomic sequencing as a research test on the brain biopsy, mNGS was able to identify the causative organism. And then finally, there were a third category of missed infections, which were low pathogen titers. And by that, these are subthreshold cases where although we did detect sequences corresponding to the causative pathogen, the sequences were below the pre-established threshold for calling it a positive. So these subthreshold detections were actually critically important, and they were discussed as part of the Clinical Microbial Sequencing Board. They include, for instance, some cases of fungal and mycobacterial infections, and we felt that this was actually important enough that now we are currently actually reporting these subthreshold cases. But notably, in this study, among the 58 diagnoses that were eventually made in infectious diseases, 13 of them were required, actually, the mNGS test, of which 8 of them had direct clinical impact. Ultimately, the highest diagnostic yield really results from combining this test along with conventional microbiologic testing to diagnose these very difficult to diagnose neurological infections. I want to give you an example of a case. This is not from the study, but this was a subsequent case. This is a patient who has chronic meningoencephalitis, a 60-year-old male from New England in the United States with a history of mantle cell lymphoma, had been on remission and had been on an antibody drug, rituximab, since 2014. So he's immunocompromised. He developed fatigue, arthralgias, which are muscle joint pains, muscle pains, and weight loss over 6 months. He was treated for Lyme disease. Initially, they thought he had Borrelia burgdorferi infection, but did not improve, and would develop kind of rapid progressive neurological illness and dementia. On exam, he had impaired arousal and attention. His cranial nerve exam, muscle tone, strength, and sensory and reflex exams were all normal. A complete laboratory workup was normal, and the spinal fluid just showed mild pleocytosis. Also, the imaging was unremarkable. And this gives you an example of kind of the difficulty of diagnosing these kinds of infections, especially neurological infections, which, again, Dr. Michael Wilson will discuss in more detail in the next talk. But this was actually an example of a very rare and unusual infection called Jamestown Canyon virus infection. This is a Bunya virus, which is now actually prevalent. There have been several cases now in New Hampshire and in Vermont, New York, and in other places in the northeastern United States. But we were able to identify this using metagenomic next-generation sequencing. And it was actually very difficult for us to confirm this result. We obtained another spinal fluid sample and then ran it for Jamestown Canyon virus PCR, but it was negative. Eventually, a serum RT-PCR the diagnosis. Although there is no FDA-approved treatment for this, the patient was treated with some experimental medications, including favipiravir, which is an antiviral, along with intravenous immunoglobulin. Unfortunately, he did not clinically improve and eventually died from his illness. But his diagnosis was made very late, about 7 months into the course of his disease. And perhaps the outcome might have been different had we run the test, the metagenomic sequencing test earlier. I want to move into another kind of sequencing test that we've developed, and this is a plasma test. So this is a test from blood, and the goal is to be able to identify bloodborne infections. But in addition to that, the expectation, the hope is that running the test from blood, we can identify kind of deep-seated infections that would be difficult to diagnose otherwise, especially because many of these critical ill patients, for instance, it's difficult or in some cases not indicated to be able, it's very difficult to get tissue from an infected organ. So one of the applications would be difficult to biopsy end organ disease, perhaps a fever in immunocompromised patients such as neutropenic fever, ICU infections, and then fever of unknown origin and culture negative endocarditis. But I want to give you another example. This is a case of a 40-year-old male who has leukemia, who was admitted with cough and fever, had developed acute respiratory decompensation, so he developed pneumonia. This was prior to the COVID pandemic, but he had then developed a kind of progression with new nodular consolidation, and it was concerning because he's leukemic. There was a concern that this was an invasive fungal infection. He had a complete workup that was essentially unrevealing and was put empirically on Zosyn, piperacillin, tazobactam, broad-spectrum antibiotics, as well as ambisome for empiric treatment of fungal infection, but continued to have increasing oxygen requirement, was not doing well, and there was actually a discussion to transition him to comfort care because he was not improving. So this was, and this is a kind of a slice through his chest CT. You can kind of see the extensive nodular consolidations that are present, mainly on the left side. But this was actually also run on our plasma mNGS test, and this was a positive case for Mycoplasma pneumoniae. What's kind of remarkable about Mycoplasma pneumoniae is that it has been known to cause pneumonia. Very, very severe infection, life-threatening infection, is actually quite rare and uncommon. And in fact, this diagnosis, again, had not been entertained, had not been considered by the infectious diseases consults. So, but we did identify, this is called a coverage map, where I'm taking the reads, and this is done in an automated fashion by the software. We're taking the sequences and mapping them across the genome of Mycoplasma. So this appears to be a bona fide real hit. And it turns out that the patient was started on azithromycin, which is the course of the indicated treatment for Mycoplasma, improved rapidly and was extubated on room air. He had also received steroids because they thought this might have been perhaps pulmonary graft versus host disease, but that was actually not continued, it was actually discontinued. And notably, he had actually received azithromycin already, a 5-day course, as an outpatient empirically. But this was really thought insufficient to treat his disease. And again, this is another example of where the potential clinical utility of implementing metagenomic sequencing, it allows you to make diagnoses that otherwise you would not be able to make. I want to move into some new technologies, and these are really exciting because they hold the promise of expanding the breadth of applications that you can get from metagenomic sequencing. So one technique that we've been working on is implementing nanopore sequencing. And so the nanopore in the main commercial device is provided by a company called Oxford Nanopore Technologies. This is a nanopore sequencer, and it's powered by the USB port in your laptop. You can actually read out the run on your iPhone or on your Android phone or other smartphone. And the idea behind the clinical nanopore sequencing is that you can be able to run metagenomic sequencing in about 2 hours. We've gotten, for instance, the library prep stage down to about 1 hour, the sample processing steps. The bioinformatics analysis can be done in real time. So as you're sequencing the data, you will be able to get real-time information and real-time results. And then ultimately, the goal is that you can make a diagnosis in 1 to 2 hours. And this is actually very exciting because it raises the possibility that we can develop this test to be truly a point of care test instead of a test that's only done by specialized laboratories. So to give you an example of the clinical utility of this test, this is my postdoc, Dr. Wang Deng, who went to Mali, the country of Mali in Africa. And here you see him actually training the Mali physicians and the pathologists here at the Institute of Virology on how to actually load a flow cell. And I have a short video here which can show you kind of the clinical utility of using metagenomic sequencing for diagnosing febrile illness cases in Africa.
We are going to sequence a sample we received from Dr. Dao at the UCSC hospital. The sample has already been extracted, the RNA library has been prepared, and we are now about to load the sample onto the MinION. The sample was collected from a patient who has been bitten by a dog, and 3 samples have been provided to us: CSF, plasma, and saliva. We have individually barcoded the samples and mixed them together to be sequenced. Wayne is now loading the sample into the MinION, and sequencing will begin shortly.
Now, the library prep step took about 2 hours, but you can see here how rapidly we can generate data. And in fact, this is in real time. I show you kind of the time graph on the lower right. You can see in this case, we were able to identify the diagnosis in roughly about 15 minutes following the start of sequencing.
Let's look at the hits file and see if anything has been detected. 7 rabies reads. There are 7 rabies reads we detected in the first 52,000.
So that was actually a case of rabies. And notably, this is a laboratory, although it's a relatively advanced laboratory, they have no capability of testing for rabies. And it happens to be a disease that's endemic in West Africa. So I want to move on to kind of the next step in terms of our endeavors. So we're working on developing metagenomic sequencing of all body fluids because one of the attractive aspects of mNGS is that you don't have to do it on just 1 or 2 body fluids. And you can actually run the exact same test on, in this case, several different body fluids. So this is a study that was published in Nature Medicine where we directly compared nanopore mNGS against Illumina, which is the traditional laboratory-based mNGS, on a benchtop sequencer. And you can see here that there are some differences. We developed what we call a dual library prep. And the idea is that you could prepare the sample one way, but you'd be able to sequence it on both the nanopore or on the Illumina sequencer. There are some advantages and disadvantages to both. With nanopore sequencing, you can get results much more quickly. With Illumina sequencing, though, you get much more sequencing depth, and you also have the ability to do many samples in parallel. So if I had, for instance, more than 30 or 50 samples, I would prefer to do it on Illumina versus if I only had like a few, like 1 to 5, I would probably choose to do it on a nanopore sequencer. But you can see here, we were able to show that with this assay that we're getting excellent performance, that essentially Illumina versus nanopore sequencing is equivalent. And we had a very high sensitivity and specificity. So the accuracy was high for diagnosis of, in this case, fungal and bacterial infections. So I want to give you an example of, this is a case from this study. This is a 2-year-old infant who had new onset seizures. So a baby, new onset seizures and a left brain abscess or fluid collection, had a history of multiple ear infections prior to this, was treated with antibiotics, but continued to do poorly, presented with new onset seizures and a wobbly gait, had decreased use of his right hand, and had headache as well. No significant exposure history, and he was admitted for workup. And so this gives you, this is an MRI scan, you can kind of see kind of the size of this fluid collection in this infant. He had a complete workup. I can just tell you that the workup was negative, and you can see here that the differential diagnosis is very, very large for this kind of infection. It turns out that we were able to actually run the sample on a nanopore sequencer, and we got an answer in about 3 hours. So this happened to be a case of culture negative Streptococcus pyogenes, or group A strep. So again, it sort of highlights the ability to not only identify unusual organisms, but also very common organisms or pathogens that we see routinely in the hospital. I want to move into some other aspects of sequencing before I end with SARS-CoV-2. One thing that we're very interested in is the use of transcriptome profiling. The idea behind it is to use the host response to be able to diagnose infections. So what we're doing here, the general idea is, imagine you have a series of images of cars in this case, and this is exactly how Google image or it probably how Baidu as well works, is that you have a series of like cars that are then then trained, and we develop essentially using machine learning, we develop a classifier by which you can then identify not only the car, but you can potentially deploy the model and identify related cars. And so I'm going to use cars as an analogy because we can do the same with infections. So this is a study where I'm taking the mNGS, CSF, spinal fluid metagenomic data. I'm identifying the viral, bacterial, fungal, autoimmune disease and other infections and illnesses. And what we do is we develop a model. We then train the model and then deploy it on a test model to assess kind of the accuracy of your model. We were able to show, for instance, that the performance is actually quite good. We're getting anywhere from 60 to 86% sensitivity, but we're still retaining very high specificity. But particularly important would be the potential use of this host response classifier to identify autoimmune disease or non-infectious etiologies. In fact, as Dr. Wilson will be discussing later, it's now thought that non-infectious causes are probably more likely or more common than infectious causes in causing encephalitis, and of which autoimmune disease, such as NMDA receptor encephalitis, might be play a large component. So the ability to be able to identify non-infectious causes by the use of human host response analysis, RNA sequencing analysis, is very promising. And this gives you another example of how we potentially might be able to, in the future, be able to diagnose infections not only by directly detecting the organism, but we can potentially identify it by their host response. This has a lot of benefit because there are many diseases, including several viral infections, for instance, where it's sort of like a hit and run sort of situation where the virus is only detectable for the first couple of days but the damage done by the virus or the pathogenesis persists for weeks. So having a test where we could look at the host response to diagnose infection would be in principle very promising. And what I anticipate in the future, that very near future, we'll be able to add host response analysis. It essentially is data that comes for free because we're sequencing the human DNA and RNA at the same time that we're sequencing for the virus or for the pathogen when we're doing metagenomic sequencing. So this gives you an example of where not only can we provide the result, which is Powassan virus in this case, as well as nocardia, which is a bacterium in this case, but we can provide kind of a human host response that can help guide management in the future. I want to end my talk by talking about how we've managed to apply many of these sequencing approaches to SARS-CoV-2 and towards helping to address the COVID-19 pandemic. Most of you are probably well aware of the circulating variants of concern, of which alpha, beta, gamma, and delta, and epsilon, which are the World Health Organization designations, of which delta is the predominant variant worldwide now. We were able to use sequencing essentially to identify, for instance, the presence of a new variant. This was in December, that was called epsilon, B1427429. We were able to show that, at least in California, that this went from 0% of cases to more than 50% of cases over the course of a few months. Currently, this particular lineage or variant has now declined, can't really be seen. It's been overcome or supplanted by Delta. But more recently, we've been able to show, for instance, again, using sequencing, that we can use it as a way to track vaccine breakthrough infections. So this is a recent study which we just made publicly available as a preprint, where we sequenced 1,373 SARS-CoV-2 genomes. And you can see here that what happens is that vaccine breakthrough cases appear to be, the infections that are seen in fully vaccinated individuals appear to be skewed towards variants that are more resistant. And this gives you an example where the unvaccinated cases, which are the lower row, really they get infections that represent the circulating variants in a community. Whereas for the vaccinated cases, we're seeing that clearly that more resistant variants are predominating in vaccinated cases. So this gives you an idea of how sequencing can be used potentially to guide public health. And we're also working on, again, in terms of the host response, we're working on ways to identify host biomarkers using sequencing again. These are RNA gene expression biomarkers that can perhaps diagnose COVID-19 and predict severity. So essentially, this is sequencing. We're just doing RNA sequencing at a very high level from both respiratory samples and from blood. And so we published a study a few months ago where we were to identify, for instance, again, a classifier that can diagnose COVID-19, that can be used to distinguish COVID-19 from other respiratory infections. We are also working on a related host response analysis classifier that can discriminate between mild and severe disease. And where I see the clinical utility of this approach, we were able to identify, for instance, specific genes that are associated with more severe COVID-19. So the idea would be that if we could develop this into a test informed by the sequencing, we would be able to screen patients in the emergency room, for instance, to determine whether or not they need to be hospitalized, for instance, if they present with severe COVID-19. So I want to end by acknowledging all of the people in my lab who are involved in this work. They're shown on the left. There are really too many to name. The members of the UCSF Clinical Microbiology Lab, UC San Francisco, including my collaborators, Dr. Wilson, who will be speaking next, and Dr. Joseph DeRisi, collaborators in public health and in other hospitals. And then finally, I'd like to acknowledge funding from the NIH, the US Centers for Disease Control, and numerous philanthropic foundations, as well as the California Initiative to Advance Precision Medicine. Thank you, and I would be happy to answer any questions.
Wow, what a fantastic talk. Thank you, Professor Chiu, for the not only novel sequencing technologies advantage and also the host response to combination to diagnose the infectious diseases. According to a time limitation, do you mind that we leave the questions to the panel discussion session? Yeah, thank you, Professor Chiu. Thank you. And I believe that all of you will have a lot of questions, want to interact with Professor Chiu. Let's have it under panel discussion session.